did not attend, we had great presentations on contaminant transport in groundwater by Dr. Ingrid Padilla, followed by environmental epidemiology by Dr. John Meeker. And those are both archived on the Protect website. This week we're really happy to have, for our third session, Dr. Rita Locke-Caruso, who is the Protect Project 2 leader from the University of Michigan. She's a professor of environmental health sciences at Michigan. And as I said, she's the project leader of the project called Pollutant Activation of Cell Pathways in Gestational Tissues. Dr. Locke-Caruso is a toxicologist with a research focus in female reproductive toxicology, and in particular, mechanisms of toxicity related to childbirth, such as premature birth. She directs the University of Michigan EHS Core Center on Life Stage Exposures in Adult Disease, and the Environmental Toxicology and Epidemiology Pre- and Postdoctoral Research Training Program. In addition to all those activities, she has served on numerous local, state, and national committees, including the City of Ann Arbor Environmental Commission, NIH Grant Review Panels, and the Institute of Medicine Committee on Understanding Premature Birth and Assuring Healthy Outcomes. And amidst all of these busy activities, she's made the time to prepare and present this session today, which we really appreciate it. And with that, I'm going to turn it over to Rita to present the third session of the Protect Academy. Rita?
Okay, thanks, Tom, for that introduction, and thanks to all of you who have called in to listen to this talk, and I hope that you learned something that can help you with your research. This is, as Tom said, I am part of the Protect Center, and at the University of Michigan, of course, we have to acknowledge the generous support from the National Institute of Environmental Health Sciences. So there are many different definitions, and we're going to start with a couple that just to orient ourselves. Environmental health sciences in public health aims to promote the health and quality of life by preventing or controlling diseases or deaths that result from interactions between people and their environment. This is taken from the mission statement of the CDC National Center for Environmental Health, and I like it because of the emphasis on interactions between people and their environment. And in toxicology's place in this public health scenario is that in environmental health sciences, we're looking at relationships between exposures and health outcomes. And toxicology has the role of defining the mechanisms that provide the links between exposures and health outcomes. This is a definition of toxicology from the Society of Toxicology as the study of adverse physiochemical effects of chemical, physical, or biological agents on living organisms and the ecosystems, including prevention and amelioration of those effects. However, for this talk, I am going to keep the emphasis on chemical agents and living organisms. So there's lots of words we use to talk about toxic substances. I'm not sure of the diversity of the audience, and I can't even see you. So I have very little idea of where you're at, but let's just give a few names for how we talk about toxic substances. The most general correct term for a toxic substance is a toxicant, and this is a broad term that means that this is a substance that can cause harm to health. Sometimes you hear the term toxin used interchangeably with the word toxicant, but toxin is actually a toxicant of biological origin. So here I show you the only rattlesnake in the state of Michigan. And so rattlesnake poison is an example of a toxin. So it would be a bee sting, for example. You've seen the term poison, and poison has a very particular connotation, and you can see it here with the skull and crossbones. So most of the time when we say something is toxic, if we use the word poison, we impute some kind of urgency to it, that there's imminent and disastrous harm potential with such an exposure, say to cyanide gas. Another general term that is often used is xenobiotic, and xeno, of course, refers to foreign, not foreigners, but anyway, foreign, and biotic to an organism. So a xenobiotic is an agent that is introduced from outside of the organism. The organism itself, the person, cannot make this substance, but rather has to be exposed to it from an outside source, and so that's also sometimes called an exogenous agent, exogenous meaning of an outside origin. So we have mechanisms for dealing with toxic substances in our bodies, and we have these mechanisms because humans have evolved in a world in which we are exposed to noxious chemicals. And humans are now introducing new chemicals at a rather rapid rate every year. So we luckily have some ways in which we deal with them. And important for understanding how bodies and organisms deal with these toxic substances is to appreciate that toxicology sits at an intersection of chemistry and biology. And as such, it depends on both the physical and chemical characteristics of a toxicant, as well as the biological characteristics of an organism. And something is not toxic, something does not cause intoxication unless it goes from exposure to a clinical symptom of dysfunction, disease, or disorder. And so it starts with the exposure. We have to be exposed to some kind of an agent but exposure isn't enough. Often you will see in the news, newscasters will often equate exposure with harm. But you have to have that exposure of that substance getting into the organism. And once in the organism, it has to distribute to a susceptible tissue, which may or may not be the site of contact. And when it gets to the susceptible tissue, there has to be a molecular reaction between that chemical and some biological moiety at the target site that results in a response at the cellular or tissue level and ultimately to physiological impairment. So the main point here is that none of these preceding steps are sufficient to declare that an exposure has resulted in intoxication until you get and unless you get physiologic impairment. And the reason is because we have something called homeostasis. And homeostasis is that ability for an organism or a cell to maintain its equilibrium in a world in which it's going to be exposed to various factors that will try to perturb it. It does this by adjusting physiological processes through either repair or compensation. And toxicology has to distinguish between these changes that are homeostatic from those changes that are pathogenic. So you have normal function, toxicant exposure, and you get some perturbation here. That in itself is not harm because there's opportunity for homeostasis through repair or compensation to return to normal function. And our bodies will try to do that. It's only when we have the abnormal function unable to be compensated or repaired for that, and we go through a pathogenic process to lead to disease, disorder, death, that we have a toxic outcome. So let's talk for a minute about exposure. In the environment, we have the main media through which we would be exposed to environmental chemicals are water, soil, dust, food, air, and they interchange as shown in this diagram. And these lead to various ways in which we are exposed through three major routes of exposure: ingestion, inhalation, and skin contact. That's all I'm really going to say about exposure in terms of contact with the media, because actually where we are evolving in the science of toxicology is towards a new paradigm of exposure, where we're relying not only on measurement of chemical substances in the environmental media in our contact with those substances, but we're also using response markers as indices of exposure. And so a biomarker, there are different representations of it, and I happen to like this one. It's pretty concise. It's an indicator of biological response to an environmental stressor that can be objectively measured. And these could be molecular, cellular, or physiologic. So an example of molecular biomarker is DNA damage. Example of a cellular biomarker could be cell death, For example, a physiologic marker can be a change in respiration. You're breathing more rapidly or your heart rate goes up. And we have the omics revolution, which is now introducing a new way to look at responses and response biomarkers to environmental exposures, and that is to do pathway analysis, looking at these physiologic and pathophysiologic pathways as they are perturbed by exposures to toxicants. And so these can include xenobiotic metabolism pathways, DNA damage pathways, epigenetic regulation, inflammation, oxidative stress, etc., and using the new omics technologies to identify signature panel responses to toxicants in these pathways using genomics, proteomics, metabolomics, computational analysis, etc. So just to wrap up on exposure, I just want to, for those of you who may not be familiar with it, what are we exposed to? Well, the CDC is doing a biomonitoring study in which they're measuring chemicals in blood and urine from a random sample of the U.S. population every 2 years, and they publish their findings. And the latest report included 212 chemicals. I think I didn't update the picture, sorry. Okay, so we said we have to be exposed to something, but that's not enough for it to be harmful. And when the next step in the pathway of intoxication is absorption. And absorption, we're going to talk about in a larger context of ADME, which is the movement of toxicants into, around, and out of the body. And the ADME stands for A for absorption, D for distribution, M for metabolism, and E for elimination. And it's important to recognize that in this toxicokinetics framework, that absorption, distribution, and elimination are dependent on the ability of these chemicals to cross barriers. They're cell barriers, the cell wall to get into cells and out of cells, and they have to cross epithelial barriers. So it's important to understand how toxicants cross these barriers. And key to understanding that is that the cell membrane is selectively permeable. And the cell membrane is selectively permeable because it has this bilaminar phospholipid membrane. So there are two layers of phospholipids depicted here. The phosphate groups are shown in blue, and they're hydrophilic because they have lots of ionized oxygens on them. So they're hydrophilic, and they're going to face the outside of the cell or the inside of the cell. And they have these lipid tails, and they're going to face inwards towards the inner part of the membrane. And so we have this bilaminar lipid membrane, and this is going to limit the movement of water-soluble compounds. It's going to like those fat-soluble compounds that are lipophilic. Now, water-soluble compounds can cross the membrane through other mechanisms. Very small ones can go through channels. These are physiologic channels, but still small ions like mercury, et cetera, can go through them. And there are carrier proteins, transporters, that can carry molecules across. But the most important mechanism for transfer of toxicants across a cell membrane is simple passive diffusion. And this is where a chemical moves simply down a concentration gradient. It'll go from one side of the cell membrane to the other side of the membrane, going from the higher concentration to the lower. It does not require any cellular energy, does not require any transport proteins. And the properties that will promote passive diffusion is lipophilicity, that love of lipids. So if something is if a toxicant is lipophilic, it's more likely to move by passive diffusion. If it's hydrophilic and very small size, then it can move through the membrane ion channels I described. And also, these large hydrophilic molecules are not going to go across the cell membrane by passive diffusion, simple passive diffusion. They're going to need another mechanism. The most common other mechanism is active membrane transport, and the kind relying on membrane carrier proteins. And what's important to remember about active membrane transport is that it's selective. These transporter proteins, they like certain molecules. They have certain chemical characteristics. and they're saturable. There are only so many of these transporter proteins sitting in the cell membrane. So once you get above a certain concentration, there's no further increased transport across the membrane. They also require cell energy, so you've got to have a healthy cell. They can transport toxicants against the concentration gradient, and this can be very important for toxicology because it means that some toxicant in a relatively low concentration in the blood can actually accumulate to orders of magnitude higher concentrations in critical cell types. And they also can transport toxicants with low lipophilicity that normally wouldn't go across the membrane easily. So that's how things get across the cell. How do things get into the body? Getting into the body, toxicants have got to go across epithelia. And epithelia are tissues that form the barriers between the inside and the outside of the body and the body compartments. So, of course, the one epithelium everybody's most familiar with is the skin, but also epithelia line the gut, line the peritoneum, line the heart, the inside and the outside of the heart. Anywhere you've got a barrier between a body compartment, you've got epithelia. And there can be pores between the epithelia that limit the diffusion between cells, and these are formed by the so-called tight junctions. And the size of the pore, the size of the molecule that can diffuse between cells is based on the molecular weight and dependent on the tissue type, on where this epithelium is and how tight those tight junctions are, because they're not the same occlusion in every place in the body. But even so, most transport, even across epithelia, is not through these pores. Most tissues have sufficiently occluding junctions that most transport across epithelia occurs by across the lipid cell membrane through one of the cell mechanisms that we described earlier. So we're just going to talk very quickly about the three major routes and skin absorption. This is a diagram of the skin. And the most critical protective component or limiting component for transfer of toxicants across the skin is this layer up here. It's called the stratum corneum. And it's basically a bunch of keratin protein that determines the thickness of your skin. And so skin absorption is modified. The major critical factor is the thickness of this layer, this keratin layer, and that's called the degree of cornification. So in some parts of your body, you have very thin skin, like at your eyelid or the skin that covers the scrotum. And at other places, it's pretty thick, like on your elbows or the heels of your feet. And anything that breaks this skin, of course, leaves you vulnerable to infection, to toxicant exposure. And this includes cracks, burns, wounds, etc. Now, the GI tract, or the gastrointestinal tract, is the tube that basically goes from the mouth to the anus. But where most of the absorption occurs, the most critical site for absorption is the small intestine. And the reason why the small intestine is the most critical site for absorption is that it has a very large surface area, about 200 meters square. And it has a large surface area because it's got this fractal almost quality to it. And so it's not a smooth lining, the tube of the intestine. It's got all these folds. So the folds increase the surface area. And then along the folds, you have villi. These are these protrusions here. So not only do you have the folds, but along those folds, you've got these villi protruding into the lumen. And not only that, but on each villus, you have these tiny microvilli. And so each villus, there's about 4 to 5 million villi, and each villus has about 2,500 microvilli. So you have this incredible amplification of surface area. And then inside the villi are these blood vessels, which form these capillary beds, and they're right next to the epithelium. So they're just waiting for stuff to transfer across that epithelium of the small intestine and be absorbed right into the bloodstream. And toxicants that get absorbed by the GI tract get delivered first to the liver, which can be very critical for toxicology because the liver is a very metabolic organ. The respiratory tract is the exposure route for things that are inhaled in the inspired air. And what's critical to understand about that is that, again, it's a tubular system. You know, it starts at the mouth and the pharynx and the trachea, and there are bronchi. And then there are these bronchioles, and they bifurcate about 16 times in most people. And each bronchiole, or tiny tube, ends up in one of these little sacs called an alveolus. And the alveolus is the working business of the lung. This is where the gas exchange occurs, where the oxygen goes into the blood and the carbon dioxide comes out of it. And it is an extremely thin epithelium. And because it's such a thin epithelium, it's very easily damaged. And that's why inhalation of particulate matter and asbestos fibers, cotton fibers, and coal dust is so dangerous and damaging to the lung, because this epithelium is so thin and fragile. And the other important thing is that toxicants get absorbed from the lungs, get returned to the heart, and then directly circulated through the body without being sent to the liver for possible metabolism. So the next step is distribution in the ADME path. And I like to remind people, especially if you're not biologically oriented, it's easy to think that blood is just a red liquid. But it's not. It's a very complex fluid. It's got cells and proteins and lipids and enzymes and other stuff floating in it. It's very complicated. And people, when they, non-toxicologists or non-biologists, don't really conceptualize well how toxicants move around in the blood. They don't just float in the blood freely. That they actually interact with these other components in the blood. So it's important that we understand these properties of blood, because blood is the major delivery system for distributing toxicants throughout the body. And so the pattern and the rate of blood flow from the site of absorption or contact with the toxicant to the tissues is very important. So there are these, blood protein binding is especially critical for toxicants. Most toxicants in the blood actually are bound to something. They're not sitting there floating as free, unbound molecules. And the most abundant blood protein that binds toxicants is albumin. And albumin is made in the liver, and sometimes we see modified responses to toxicants because of liver toxicity. So patients with cirrhosis, for example, who might not be making as much albumin, have lower albumin concentrations, have different toxic responses. And because this protein binding can modify absorption, it can help move compounds across an epithelial barrier into the body, because once the toxicant binds to a blood protein, that concentration gradient is maintained, because the concentration gradient is based on the free molecule of the toxicant. And likewise, it modifies distribution and excretion for the same reason. Once bound to a protein, it will be excreted differently and distribute differently. And toxicants and endogenous substances can compete for binding to proteins, which can sometimes be problematic. So, for example, we know that many of the halogenated toxicants, like PBDEs, will bind to some of the hormone-binding proteins in the blood. We think that contributes to some of the health issues. In addition, for distribution, toxicants can bind to tissue proteins, and that can be a way in which certain tissues become like sponges and soak up particular proteins. And a good example of that is the protein metallothionein, which is induced, especially in liver and kidney, with exposure to certain toxicant metals, in particular metals cadmium, mercury, and lead. So I already mentioned blood flow is important. Toxicants will tend to concentrate initially in those tissues that have high blood flow, but then with time, they can redistribute to other compartments. So you have to consider time and blood flow in any models of distribution. And as I mentioned, active transport can concentrate toxicants at a particular target cell. Common storage sites in the body are the liver and kidney, in particular because they have a high rate of blood flow, and they also express higher concentrations of certain toxicant binding proteins, like the metallothionein example I gave you. Fat is also a common storage site for lipophilic toxicants, but these toxicants are released during lipid mobilization. So, for example, during fasting, starvation, during pregnancy, there's terrific lipid mobilization during pregnancy and lactation. And what this means is that the fat can actually be a depot for these lipophilic toxicants, and you don't have to necessarily have current exposure to lipophilic toxicants during pregnancy, for example, in order for the fetus to be exposed, because simply the fat mobilization that's occurring as part of normal pregnancy is going to be releasing those toxicants into the blood, elevating circulating levels of those toxicants, and delivering more of them to the placenta. Bone is another storage site for certain toxicants. The bone is continuously remodeled, and during the remodeling, certain toxicants have an affinity for some of the components of blood, and they can be then incorporated there and stored there. Lead, strontium, and fluoride are some good examples of that. And then during processes of bone demineralization, as well as during just regular remodeling, but especially during bone demineralization where there's more coming out than going in, such as osteoporosis, pregnancy, and lactation, the bone serves as a depot or a source of exposure. And this has been well shown for lead, that women who are exposed to lead earlier in life but don't have significant exposure during pregnancy nonetheless can expose their fetuses to lead through release of lead from their bone source. And we're going to skip over metabolism for a minute and go to elimination quickly. There are three major routes of elimination: the kidney, intestine, and lungs. And the kidneys excrete mostly the small, unbound, water-soluble toxicants, or ones that are bound to small proteins, via filtration into the urine. But there are also these carrier-mediated transport functions, these transporters. And one important factor about them is that these transporters can be saturable. And if your toxicant is dependent on transport into the urine from the blood through one of these transporter proteins, then above a certain concentration, you may start accumulating excessively high concentrations of the toxicant in the blood, whereas low concentrations you can handle because they get excreted. The GI tract is the route of excretion for material that's not absorbed by the stomach or intestines, as well as by substances that are metabolized in the liver, sort of depicted here, although I've never seen a liver that looked like that. And then it gets shunted down through the gallbladder and the bile into the intestines. And the lung is the route of excretion of gases that are lipophilic and have low solubility in blood. So many of you may have had the experience of nitrous oxide at the dentist's office. You inhale it, and you get very quick neurological response. But also, as soon as you take the mask off, you're very quickly restored to your normal function, and that's because that is a gas that has low solubility in blood, and so it's rapidly eliminated in the air. And there are minor routes of excretion. The saliva, hair, and nails are notable for their potential use to monitor exposure. So this is a very non-invasive. It's pretty invasive to stick a needle in somebody's arm. A lot of subjects and populations are not too keen on that. And it can be problematic to collect urine samples, but collecting saliva, there are these little saliva packs you can use. You just have them stick them in their mouth, under their tongue for a minute or so. Hair and nails have been shown to be quite useful. And milk, breast milk, is also an interesting medium, a route of excretion. There are some compounds, there are some chemicals like DDT for which excretion in milk is the only significant route of elimination. And that's because milk has a relatively high fat concentration. It's about 4% like fat, like cow's milk. And so these lipid-soluble compounds that are circulating in the blood will distribute into the milk. And now we're going to go to metabolism. And I got to tell you, metabolism is lectures, multiple lectures. So there's no way, I'm just going to give you a little bit of taste of why we have to consider metabolism so much in toxicology. First, it's important for the elimination of toxicants. Lipophilic toxicants are are easy for us to absorb into our body, but difficult for us to get rid of because they're not going to be excreted in the urine. They're going to be absorbed from the gut. How are we going to get rid of them? And we need to before they can accumulate to hazardous levels. Well, we do this because we get rid of it, and we're protected because we've evolved mechanisms for metabolizing them. So most lipophilic toxicants actually are transformed or metabolized into these hydrophilic or water-soluble metabolites that can then be excreted. And so here's just a diagram showing how metabolism modifies compounds to forms that can be more toxic. In the top pathway, this is called bioactivation, where metabolism will change the chemical structure of this toxicant. It'll add electrons, take away electrons, add oxygen, add even other larger moieties, and bioactivate it, forming an ultimate toxicant, which then exerts its impact on the cells and tissues. Or metabolism can convert that molecule into something that's less harmful. Sometimes it even goes this way or this way. But we try to simplify it here to make the point that metabolism is two-sided. It can be beneficial, protecting the organism from hazard, and it can be hazardous, bioactivating a substance to something that's more dangerous. So let's take an example of ethanol metabolism, because I think most of us are familiar with alcohol. So we're going to start with, I should have said ethanol. I should have said methanol, because ethanol is the next thing I'm going to show you. But methanol here is actually a toxic compound. This is sometimes ingested accidentally or for suicide attempts. But it gets converted by the enzyme alcohol dehydrogenase. It gets metabolized to formaldehyde, which is not a nice compound to have in your body. It's very reactive, but formaldehyde then gets converted from aldehyde dehydrogenase to formic acid, which is even nastier and leads to blindness. Alcohol, or ethanol, the alcohol that we like to drink on Saturday night, maybe even on Father's Day yesterday, ethanol also gets metabolized by the same pathways. Alcohol dehydrogenase to form the acetaldehyde, and then the acetaldehyde by aldehyde dehydrogenase to form acetic acid. And acetic acid is something we like to cook with a lot and put in our salads. It's vinegar. I forget what the percentage is, 4%? Acetic acid is vinegar, something like that. I looked at my students and they gave me no high sign. Okay. Anyway, so acetic acid obviously is something that's not harmful to us. And what makes you sick from drinking too much is this acetaldehyde. And in fact, a significant percent of Asian people have a polymorphism in the gene for the aldehyde dehydrogenase that makes them slow metabolizers of acetaldehyde. And those are the people who turn pink when they drink too much alcohol. They have two glasses of beer, nothing that would make someone without that polymorphism ill, but they get ill faster. And the antabuse, which is used as sort of an extreme measure for treating alcoholism, inhibits this enzyme and makes people sick because the aldehyde is what makes us sick. So why do I show it on the same slide with methanol? And that's because actually giving ethanol is used clinically as a way of treating methanol toxicity. And that's because the ethanol will compete with the methanol for alcohol dehydrogenase, and the acetaldehyde is much less toxic than the formaldehyde. And so people who come into the ER with methanol, oh. I woke everybody up, are treated with ethanol. Another example I'm going to give is acetaminophen, and this is Tylenol. And Tylenol is normally under low enough concentrations, humans will perform this metabolic conversion here, which adds this moiety. They don't show you the chemistry of it. It's a rather large moiety, glucuronide, to form this metabolite, which is not toxic and it's easily excreted. And that's how we normally handle acetaminophen so we don't get into trouble. However, if there is too much acetaminophen, as in a suicide attempt, or if this step is inhibited or is low activity for various reasons in a person, then you don't form enough of this or this metabolite. You get instead favored metabolism to form this very dangerous metabolite here, which is the ultimate toxicant, which then reacts with proteins, nucleic acids, and lipids to form toxicity. And cats, for example, don't have this enzyme, glucuronidation, UDP glucuronidase, and so if they get exposed to acetaminophen, they get very sick or die. So don't give your cat acetaminophen for a headache. I'm so glad you laughed. Somebody laughed. So there are modifiers of metabolism. Lots of things can modify metabolism. We just gave an example of species. We also gave an example, I hear previous exposure, but also concurrent exposure with ethanol and methanol, but also gender influences it. I mean, there's a reason why women show more symptoms of alcohol effects with the same dose than men. It's because there are gender differences in how we handle alcohol, strain differences, species differences, genetic variations, we talked about that, age. Infants, for example, have very different metabolism, newborns than older children and adults. Your physiologic state during pregnancy, pregnant women are really different, disease states, etc. So we talked about basically these three: the exposure, getting exposed to something nasty, getting it into the body, and getting it to a sensitive site. And the other three really have to do with responses at the site. And so I'm going to use that as a segue to cover dose response, which is considered one of the major areas of toxicology. I think if I didn't talk about dose response, someone would say I didn't do a proper job in introducing you to toxicology. So this lovely fellow here is Paracelsus, and he's rather famous in the toxicology community. Paracelsus was a physician who lived a long time ago, and he was treating his patients with substances that were known to be harmful. But he was successfully treating them. And a good example of that, I don't remember if Paracelsus actually used this, but a good example of that is arsenic was the original effective treatment for syphilis. And now we say, oh, arsenic, oh my God, poison, right? We put poison on arsenic. But it actually could be used and was used to treat the disease. And so he was called by the Academy to defend his use of deliberately giving poisonous substances to his patients. And in his defense, he said, what is there that is not a poison? All things are poison and nothing without a poison. Solely a dose determines a thing that is not a poison. And this has been widely interpreted and extended from the situation in which he was in for treating patients who were ill, possibly dying, to exposures to chemicals for which there may not be any benefit at all, to industrial chemical exposures, environmental chemical exposures. So, but dose does, dose makes the poison is true to an extent. The dose of something determines whether a chemical will be beneficial or poisonous. In these cases here, with aspirin, we have a beneficial dose and we have a toxic dose. With vitamin A, we have beneficial dose, but you can get too much vitamin A and get sick or die. Even oxygen, which is necessary for life and is good, necessary for life in incubator situations decades ago, babies were exposed to too high levels of oxygen and experienced oxygen toxicity, most notably blindness. So why do we care about the dose-response relationship? Well, it identifies the lowest effective dose or the threshold dose at which we'll see a response. It helps to support causality between exposure and observed effects in observational studies, such as epidemiologic studies. and it allows assessment of relative toxicity. So we get to compare toxicants by saying, you know, it takes a lot more of this compound to cause the same effect as this other compound. So if we have to choose between these two pesticides, we have a way of comparing which one is more dangerous. And then it's also used to establish exposure standards or criteria for exposure by through this knowing where the lowest effective dose is. So we measure dose various ways. We can talk about exposure dose, how much is in environmental media like water or air, how much we actually give to somebody in an administered or applied dose. What's the internal dose, how much actually gets into the body. How much is at the target tissue, so what's the target organ dose? Or sometimes we talk about total dose, the sum of all doses in case there's multiple exposures. And we measure dose different ways. We might just talk about how much of it. You got exposed to, you know, so many milligrams of whatever. I don't want to say arsenic, you'd be dead. We can talk about exposure relative to body weight, because the same amount, you know, in the air for him or her is going to have a different exposure if we take into account body weight. Or we can talk about it with respect to time. And then we can measure responses two basic ways. One is a quantile all or none response, where we talk about what's the proportion or the percentage of subjects or samples that respond at each dose. So an example with this would be how many people develop hypertension, you know, high blood pressure. So you have a clinical definition. If the blood pressure is above a certain level, they're hypertensive. Or you can talk about graded responses within each individual subject, where you're measuring the magnitude of individual response at each dose. And so this would be, what is the blood pressure? You know, how many milligrams of mercury is it versus how many people are at it. And we do have response variability. So this is an idealized bell-shaped curve for the lowest dose that initiates response. And the point I want to make here is that most people fall somewhere in the middle here. This is the majority of individuals. But with any exposure, you have people on the tail end at the low dose who are susceptible, and you have people at the high end who are resistant, showing that we have response variability. When we talk about those response relationships, we will talk about whether they're effective dose. We might call it an ED dose, or a lethal dose, LD, a toxic dose, or a therapeutic dose, a TD, or an inhibitory dose, ID. There's just terms you might come across, and that's all they mean. Those response data are usually plotted as cumulative dose response curve, with the dose on the x-axis, the response in percent on the y-axis, and it often takes the shape of a sigmoidal or an S-shaped curve. And the data then can be expressed as the proportion of response at a given dose. And the most common one you might be familiar with is the LD50. And the LD50 is the dose, then that elicits a 50% maximal response. An LD20 would be the dose down here, maybe around 15. That elicits a response in 20% of the population. An LD90 would be, say, up here, maybe 25. It elicits in 90% of the population. That's all it means. And we describe the curve. Certain points on the curve of the dose response are important in risk assessment. So we talk about a NOEL for the no observable adverse level, a SNARL, which is my favorite, the suggested no adverse response level, a LOAL, lowest observable adverse effect level, and the maximum effective dose, which is the dose at which there's no further increased response. And those are shown here. Here's the NOEL, no observed adverse effect level, LOEL, the maximum effect. So let's take an example with acetaminophen, Tylenol again, because it's something most of us are familiar with. An overdose of acetaminophen is very toxic to the liver. That's the target organ. In fact, it's the leading cause of acute liver failure and liver transplants, leading to about 400 deaths annually, with 20–25% of the deaths from acetaminophen overdose being unintentional. And that's why there's a lot of push now to label everything that has acetaminophen in it, with it. So here's an example of alcoholics defined as drinking more than 3 drinks per day. are at a greater risk for toxicity following an overdose of acetaminophen, and it's because there's a shift in the dose-response curve for acetaminophen in alcoholics. So, and actually the other point I want to make here is that there isn't a big margin of safety here for acetaminophen. And so some people say that if acetaminophen were introduced today, it would be prescription because there's such a little margin of safety. And you can see that, in fact, individuals are very near the toxic dose. So they get shifted. Now, the previous examples given were based on an increased response occurring with increased dose, but that doesn't always happen. And in fact, there are more and more examples of nonlinear dose-response relationships, and this is so-called hormesis. And these occur if you have a stimulatory or a beneficial effect at a lower dose, but an inhibitory or an adverse effect at higher doses. And these form J-shaped or U-shaped or inverted U-shaped dose-response curves for outcomes. They're relatively common with things like nutrients, where you might need a little bit, but you need more to be beneficial, and you have too much, like the vitamin A example. You need some vitamin A to be healthy. You don't have any, you have too little, you get ill. You have too much, you get ill. And they're also seen with hormonal substances. And another example of that would be estrogen. You know, women need some estrogen. Too much of it, though, is not healthy either. So these hormesis, nonlinear dose-response curves, and especially the first example where you have a stimulatory or a beneficial effect at a lower dose but an adverse effect at a higher dose, has led actually to some toxicologists to suggest that a little bit of this toxicant might actually be good for you. Why are we regulating exposure to some of these toxicants so harshly? And so this was a very nice article. That it's from 2003, but I haven't seen anything quite like it since. And took this from Nietzsche, the philosopher Nietzsche. What doesn't kill you makes you stronger. Talking about how this hormesis is revolutionizing risk assessment, but also has implications we need to think about before we start applying it. So we do know individual biological factors modify susceptibility, and that's shown by the variability in response that we see in a dose-response curve. If we didn't have variability, we wouldn't need a dose-response curve. And some of these modifiers can be biologic, like metabolism, genetics, gender, age, disease, physiologic state, and some of them can be behavior, diet, prior concurrent exposures to substances. And so we come to the point of Paracelsus being oversimplified. When someone tries to make the argument that solely a dose determines whether a thing is toxic or not, it's not true, because it fails to take into account all of these and more factors that impact on biological susceptibility. So it ignores individual biological susceptibility. Not everyone's going to respond to a toxic substance the same way or under the same conditions the same way, under different conditions the same way. I'm going to talk just a little bit about some of those responses that are seen and what the toxic targets can be anything from molecules like hemoglobin is a toxic target for carbon monoxide, through cell structures like mitochondria for cyanide, to tissues like pesticides, certain organic phosphate pesticides for nervous tissue, to organs like ethylene glycol for the kidney. So it can occur at many different levels of organization in the body. And there's no way I can really talk a lot about these different toxic targets and how they do it. So I've just handpicked a few examples. So most of you have probably heard the term endocrine disruption. And I'm going to show you two examples of how toxicants can cause endocrine disruption. The first one is through toxicant activation of cell pathways, where the toxicant interacts with a receptor or a hormone. And in doing so, it didn't. It was supposed to do something it didn't do. Okay, anyway, and in doing so, these pathways now turn on and are activated. And these pathways, through these different signal transduction pathways in the cell, will lead to changes in gene expression and protein expression, or modification of protein function. And so the cell now is behaving differently. Alternatively, you can have toxicant inhibition, where you normally have some hormone. We need thyroid hormone, we need estrogen hormone to help us be healthy. But the toxicant comes along and either kicks off that hormone or competes with that hormone for binding to the receptor, either kicks it off or prevents it from binding. And this now blocks these pathways from happening, so you don't get the gene expression for gene expression and proteins expression, or you don't get the proper protein function. That's just an example of endocrine disruption. Another common pathway of toxicity is oxidative stress. This is a very complex pathway, but basically what it is about is making molecules that are highly reactive, that include the oxygen molecule. They're very reactive, and so they start reacting with these macromolecules in the cell, lipids, membranes, nucleic acids, your DNA, proteins like channel functions, etc. It wreaks havoc on the cell. So there's some basic mechanisms of oxidative stress, including metabolism. When xenobiotics get metabolized, they can form molecules that redox cycle through metabolism, so they form a molecule that can accept and donate electrons very quickly and add those electrons to oxygen to form this very reactive superoxide molecule. Sometimes metabolism of xenobiotics just leaks electrons off. That helps to form superoxide. There are various conversion pathways to different types of reactive oxygen molecules. And if you have some kind of a exposure that leads to depletion of protective molecules, in this case glutathione or GSH, that also can lead to this oxidative stress, where you have too many of these reactive oxygen molecules now floating around in a cell, reacting with macromolecules and causing havoc. And the reason why we care about it is because it's been linked to a lot of different outcomes, adverse outcomes in many different organs and systems, as shown here. So DNA damage is also, oh, my things didn't go. Anyway, I'll point to them. So DNA, my arrows are all in the wrong place, except for that one. But DNA has lots of atoms that have extra electrons, and has polarized atoms that make them more vulnerable, more reactive with exogenous compounds. And so this is the reason why DNA is susceptible, for example, to damage from reactive oxygen species, like hydroxyl radical, or other toxicants that have reactive sites. And there are various different types of DNA damage that can occur, from breaks in the DNA, they can be single-sided or double, to actually the chemical forming a cross-link between the two strands of the DNA, to actually these where you're actually losing parts of your base, or causing single breaks, or causing binding with proteins around it. So there are various mechanisms by which we can have toxicant damage to the DNA. And and we get exposed to DNA damaging agents all the time, and UV radiation is a great example of that. And the reason why we manage to live so long is because we have DNA damage responses. So people who have certain problems with their DNA repair pathways, like individuals with the disease xeroderma pigmentosum, don't live very long unless they take extraordinary measures to protect themselves from UV light. But most of us have these mechanisms where we can protect from DNA damage from various stimuli, whether it's from metabolism or UV light or ionizing radiation like X-rays or chemicals. And they include things like stopping the cell from dividing, so-called cycle checkpoint, to give time to repair. They activate certain programs to induce DNA damage repair mechanisms. We have enzymes resident in most of our cells for repairing damage to DNA of all kinds. And if all that fails, our cells could trigger a cell death through apoptosis to kill the cell. And all this is to protect us from having cells that carry errors in the DNA. And we worry about the cells surviving with damaged DNA because there are serious adverse health consequences such as tumorigenesis. But DNA damage also can lead to other problems, whether it's through cell death or through mutation of the genetic code that include infertility, embryo lethality, birth defects, aging, and tissue atrophy. So the last mechanism I'm going to just briefly discuss is epigenetics, and this is a newer mechanism of consideration for the toxicology field, because this is DNA damage is actually probably the most widely researched, heavily researched mechanism in toxicology. Epigenetics is distinct from that because it involves looking at stable, heritable changes that do not require alterations in the DNA sequence. And so what this means, it has to do with markings of the DNA bases, where the DNA bases themselves remain unchanged. The sequence of DNA bases, the genetic code remains unchanged, but moieties have been added to, in this case, a methyl group, has been added to some of those bases, which then changes how that chromatin behaves around those bases. And the major epigenetic mechanism I'm going to talk about for just a couple minutes here involve control of chromatin condensation. So you're probably all familiar with chromosomes, and when people talk about chromosomes, they usually show them in their most highly condensed state, where you can see a shape, such as this X shape here. But within each of these arms, here's the little cutaway here showing you the end tip of it. What it really is very tightly wound up coils of DNA that's wound on itself and wound on itself again. And if we pull some of that out and look at it, what we see is that here's our DNA strand, a single strand of DNA. It gets wrapped around with proteins of the chromatin, histones, to form these nucleosomes, like beads on a string, which then get coiled even more tightly and eventually condensed into this chromosome. Well, how tightly this chromatin is coiled determines whether those genes in that area are expressed or not. In tightly coiled chromatin, the genes can't be expressed because the enzymes can't get in there to form the messenger RNA. So the molecular mechanisms of epigenetic inheritance, the main one is DNA methylation. Histone modification is also part of that because it helps to signal whether there will be methylation or not. So the methylation, when you get a methyl group added to this one base, the cytosine base in DNA, that tends to occur in so-called islands or areas of the genetic sequence where you have lots of these cytosine guanine base pairs. And this methylation, this addition of a methyl group to the cytosine is not a mutation because it doesn't change the genetic sequence. What it does do is it changes the coiling and the packaging of the DNA, the chromatin, which then determines transcription or whether it is expressed or silenced. So DNA methylation is usually associated with gene silencing, and its pattern is both stable and heritable across, can be stable and heritable for certain genes across cell divisions and even generations of people. So that's a rapid romp or a quick two-step through toxicology. I hope that you have enjoyed it and learned something. I strongly recommend a portal for those of you who want to learn something more. and it's Toxtown. Toxtown is at the National Library of Medicine site of the NIH. And in Toxtown, this isn't the active site, but I don't know if I can go to it. But in Toxtown, you can click on anything. We can start with the town. And you do your mouse over, so you hear it honk. And yeah, there's the school bell. And you can see what's of concern in the school. Oh, water fountain. Let's see. And, you know, there's different things. Particulate matter from air contamination. Mercury. Oh, that's from science labs where they ask students. They start showing students how liquid mercury behaves. Lead in the water fountain. And if you click on any one of these, and they also have it down here, but say we click on lead, what is lead? We can get a general description. You can get something on lead poisoning. You can go to the CDC. You can go to the NIEHS, EPA.
Rita, this is Kristen. Sorry to interrupt. I'm not sure what you're showing us now, but I think it's outside of your PowerPoint presentation.
Oh, thank you for telling me. I'm going to stop right now then. Just go to Toxtown. It's fun.
Okay, thanks.
I don't know how to get rid of this. How do I get rid of this? Okay. Okay, I'm so sorry. Thank you. Kristen, I would have been going on for a while. Okay, I really recommend Toxtown. Go to it. Have fun. It'll take you from grade schools, or I'd say probably high school level, all the way through scientist level of information. You can really dive down into a toxic substance through this portal. So it's great for people, especially who are not toxicologists. And I'm done. Thank you very much.
Thank you, Rita. And we do have a couple of questions offline here that I'm going to ask kind of in the order they came in. And if anyone else online has questions that they want to chat to me, again, just as a reminder, you can float to that green bar at the top of your screen and open a chat box. Rita, the first question that we had was from the NU conference room. It seems that you didn't talk about cancerous response to toxicants. Is this under DNA response, or is it a totally different response?
Cancer can result from multiple different mechanisms. It's most often thought of as a response to DNA damage, and that is a very common mechanism. Causing mutations to the DNA are linked to causing cancer, particularly mutations as well as changes in expression of some of the proteins that control the DNA damage response, so cell cycle a checkpoint, and DNA damage repair and apoptosis are controlled by a particular protein called P53. So P53 can be regulated by mutation to P53 or by changes in expression, and that's found in most cancers. Oxidative stress is important for carcinogenesis also because those reactive oxygen species, especially the hydroxyl radical, damage DNA, cause mutations. And endocrine disruption is a non-genetic, epigenetic type of mechanism of carcinogenesis. So, for example, estrogen, exposure to estrogens are associated with various cancers.
Okay, thank you. There was one other question. NIEHS is talking about the exposome. Let me just get up here. How is this related to toxicology?
Well, the exposome is really something quite different from what I talked about because when they talk about the exposome, well, I don't know. I can't say I've read everything that has lately been written on it, but the initial discussions and descriptions of the exposome were very much sort of engineering oriented, which is trying to understand and trying to get some kind of a way to measure and monitor multiple exposures in real time and over time in populations. Because when we, one of the biggest challenges in environmental epidemiology is exposure. And I didn't go into it here because it's not really you know, John talked about epidemiology, I talk about toxicology, but the ability to know what people are exposed to and how that exposure links to disease is really the major shortcoming, the challenge in environmental epidemiology. And you got to realize, especially something like cancer, for example, where you might have a 25, 30-year gap between exposures. You know, how do you do those measurements of exposure? I think that the exposome discussion has now expanded, but I'm not 100% sure, to include some of these response markers. But it's sort of a little different from the original discussions. The original discussions were really about how do we measure all this stuff that we're exposed to.
Thank you. And then another comment, very nice workshop. And then question, is there always a direct relationship between what is excreted, let's say in urine, and the concentration of a toxicant that enters the body?
Great question. The answer is no. So for two reasons. One is that you can have multiple routes of exposure. So if you're only looking at one exposure route, you may be missing others. And the other is that many of the mechanisms involved between entry into the body and elimination of the body are not going to be linear. So for example, you're going to have saturable kinetics processes in terms of maybe elimination or of metabolism. So it's not a direct one-to-one.
Thank you. And then another comment, excellent presentation, thank you. I'm looking forward to sharing the archived version via our website. And then another comment, excellent presentation. I took toxicology in grad school about 25 years ago. I wish Rita had been my professor. I might have changed my research focus. Thanks.
Oh, thank you. That's so nice. You made my day.
There is another question. Can you show the slide that shows the steps to toxicity? It starts with exposure, then entry to organism. And related to this question, Rita, would we be able to make your presentation slides as well as your WebEx available to the online via the website?
Yeah, I think so.
Okay.
Let me see if I can find this one quickly before I make everyone sick. Is this the one? Can you see it?
Yes, we can see it. And I think that's what the person's talking about, that they can.
Uh-oh. Where'd it go?
Whoops, it was the one.
Where'd it go?
Maybe you have to do view slideshow from that slide, from current slide when you click on the slide.
I don't know.
Slide number 8. Yes, there you go.
If I click that? That's what I clicked and it did something bad.
If you go to slide.
Oh, she did it.
Oh, okay.
Iman, the one.
Thank God for Iman.
Yes, yes, the wonderful Iman.
And then we just had one last question. Could Rita mention something more about environmental health and reproductive outcomes?
That's pretty broad.
Yeah, so if that person has any more specific.
I'll start talking, but if they want something more than what I'm saying, they'll have to clue me in.
Okay.
So I could go on forever about environmental exposures and reproductive outcomes because that's what I study, and I do teach an undergraduate course in toxicology, and it seems like every 9 out of 10 examples are always from the reproductive system. But we can even just look at this paradigm, exposure. So I mentioned that the scrotum has a very thin epithelium, and it's a good reason why men who work with chemicals really ought to wear chemical aprons if they're going to be if there's any danger of slopping onto their groin area, because that epithelium offers no barrier. Let me see. I don't think that's what that person's asking, though. Reproductive outcomes probably is looking at physiologic impairments, maybe with preterm birth. I don't know. But the mechanisms for reproductive outcomes are, I think the major ones are going to be the endocrine disruption and the oxidative stress and inflammation, which integrates with oxidative stress, but I didn't give as an example. But I think those are major mechanisms by which environmental chemicals activate pathways abnormally. And because they're activating pathways abnormally, you're going to get led into abnormal responses. And then I guess DNA damage for infertility and subfertility would also be important.
Thank you, Rita. Thank you very much. And thank you all for attending. As I said, this will be available on our website, the recording of Rita's presentation. And we sent a brief survey after the presentation, so we'd really appreciate if people would complete to let us know what they thought of the Academy seminar today, and also any suggestions they have. And our next and final Protect Academy webinar will be on Thursday, presented by Dr. Akram Al-Shawabka, and that will be on, I just lost my thought process, remediation, excuse me. And we hope that you all will plan to join that. I will also send a link in the email that I send out following this so that you can sign up if you've not already. Thank you very much.
She was really good. Yeah, she's a good teacher.